Historically, the primary interest of ovarian biology was studying the egg.
What's been less well appreciated is that the environment that supports that cell also changes with age. There's this term in the aging field called inflammaging. That's the idea that inflammation really increases with age and is actually a really prominent feature of aging tissues. We used ovaries from 14-month-old mice. That corresponds to a human age in the mid to late 30s. Those mice were quite advanced in age relative to other studies that had looked at ovarian aging. With age, what we find is that there's this massive infiltration of immune cells into the ovary.
Immune cells were beginning to signal more and more with the granulosa cells, which was a surprise because I don't think in the field it's typically thought that those two cells communicate very strongly.
These support cells are taking on these more inflammatory signatures and inflammatory signaling. That means that that oocyte is not getting the same kind of support that it did in the younger ovary. One of the things that we speculate based on the signatures that we saw in this study is that those support cells for the oocyte are really changing their identity with age. One of the amazing things about the aged ovary is that the tissue morphology changes incredibly. It's almost unrecognizable as an ovary, and many of the cell types are changing. So it's really important to observe those changes in the natural tissue environment. So when the spatial transcriptomics technology became available, we saw this amazing opportunity to integrate it with our single cell technology. So with single cell methods, you can get really deep information about each cell type. And with the spatial method, you get that information about how the cells are positioned relative to each other. But you get less deep information about any individual cell. We integrated these two to get deeper, more holistic information about the aged ovary.
The big picture here is that with age, the female reproductive system begins a functional decline. And this functional decline is becoming more and more relevant in modern times because the age at which women are deciding to start their families is increasing. And so because of that, more individuals are experiencing the negative implications of reproductive aging.
And so we really need to understand how our fertility relates to age. And what we hope is that in the long term, this kind of information and other studies will help us prolong fertility for women.